Good morning, dear faculty members, research scholars, and students. Welcome to the day 6 of FDP on emerging research trends in pharmaceutical sciences. Thank you, dear participants, for your interest and your positive feedback on this faculty development program. I request Mr. Anusha Madam to introduce our today's speaker, Dr. Gananadhamu Sir, Associate Professor, National Institute of Pharmaceutical Education and Research, Hyderabad. And today's topic is in silico prediction of drug degradation products and their toxicity.
Good morning, everyone. It is my great pleasure to introduce our speaker for today's session of our national level 1-week e-FD on emerging research trends in pharmaceutical sciences. Dr. S. Gananadhamu Sir, a renowned academic and Sir is currently serving as an assistant professor at the prestigious National Institute of Pharmaceutical Education and Research, NIPER, Hyderabad. Sir holds a B Pharmacy degree from KVSR, Siddhartha College of Pharmaceutical Sciences, Vijayawada, and M Pharmacy from Andhra University, and Ph.D. in Pharmaceutical Sciences from the same university. With over 22 years of professional experience, Sir has worked in both academia and industry, including Torrent Research Center and various academic institutions. Sir core research interests include analytical and bioanalytical method development, drug degradation chemistry, impurity profiling, stability studies, and herbal drugs, chromatography and mass spectrometry techniques such as HPLC, UPLC, GC, LCMS, GCMS, and more. Sir has guided 6 PhD scholars and over 70 postgraduate students. Sir has successfully completed major research projects funded by DST, SERP, and DBT, and has over 100 research more than 1030 citations and an H index of 19. Sir is the recipient of several honors, including the APP Young Talent Award, and is a life member of professional bodies like APTA and the Institute of Chemists, India. Sir also serves as a reviewer for many high-impact. S. Gananadhamu Sir will be delivering a session on high silico prediction of drug degradation products and their toxicity. We are truly privileged to have him with us today. Let us welcome Dr. S. Gananadhamu Sir.
Thank you, Dr. Anusha Madam, for introducing me. Now I am sharing my screen. Can you see my screen?
No, sir, it is not visible.
It's not visible?
Yes, sir.
1 minute, sir. Once again, I'm sorry. Is it visible, sir, now?
Yes, sir, now it is visible. Please hide that icon, sir. Stop sharing icon.
Good morning, all the faculty who are attending.
Sir. Please share your screen, sir.
I shared, sir.
While you are clicking on hide button, you are clicking on stop sharing.
Okay. Sorry. Once again, I'm sorry.
No issues.
One minute, sir. Sorry. Now, can you see my screen, sir?
Yes, sir. Yes, sir. Yes, sir, it is visible. Just right.
Now, is it okay, sir?
Yes, sir, it is now perfect.
Yeah. Good morning, all. Good morning, all the respected faculty members attending from different parts of the country for this 1 week faculty e-faculty development program. First of all, it is a very privilege for me, honor for me for delivering this talk. I thank management of Vasantidevi Patil Institute of Pharmacy, Kodoli, Maharashtra, for giving me this opportunity. I also thank Professor Shyam Sundar sir and Dr. Laxman for inviting me and giving me this opportunity. Since last 1 week, from Monday to Friday, different experts they shared their expertise in various areas of this drug development and research. Today, I'm going to talk about in silico tools for prediction of drug degradation impurities and their toxicity. Before going to this, what are these degradation impurities? So these degradation impurities, they are also called as degradation products. These degradation products are nothing but they are the unwanted chemical substances which formed from the drug substances during the storage. It may be due to interaction with the light, temperature, humidity, etc. Generally, when the drug product is developed, the care is taken so that they use a pure active pharmaceutical ingredient excipients. But after formulation, this degradation product may generate because of exposure to various storage conditions. Even small amount of this degradation products, sometimes they have toxicity, or otherwise, as the drug is degrading, so the amount of drug substance becoming less in the formulation. So because of that, the patient may not have the enough therapeutic activity, or otherwise, because of presence of these degradation products, they may have exposure to toxicity. Hence, understanding and controlling of these degradation products is crucial for maintaining the efficacy and safety. So for this, generally, pharmaceutical manufacturers, what do? They do stability testing. Before releasing the product into the market, they do stability testing. Stability testing means stability, it is the capacity of drug product to remain within established specifications of identity, quality, purity in a specific period of time. If you see any drug product, it will have a label, expiry label, manufacturing date and expiry date. Mostly some 3 to 5 years expiry date they give. So during this expiry day period, so the drug identity and its purity, it will be maintained. It should be maintained. So then only the desired therapeutic benefit can be obtained. So for this, various regulatory guidelines are there for stability testing. For stability testing, entire world, it is divided into some 4 zones. Zone 1, Zone 2, Zone 3, Zone 4. Here you can see the countries, different countries in Zone 1, Canada, Germany, European Union, Russia. Their temperature is 21 degrees centigrade, relative humidity 45%. Similarly, Zone 2, Australia, Mexico, Japan, US, China. Zone 3, 30 degrees centigrade, hot, 35% relative humidity, very dry, hot and dry area. So here Iraq, Jordan, Botswana, Chad, these countries come in this. And another Zone 4, this Zone 4, it is once again, it is divided into Zone 4A, Zone 4B. In Zone 4A, hot and humid, that region is called as hot and humid. 30 degrees centigrade, 65% relative humidity. It is typically seen there. Bangladesh, Sri Lanka, Kuwait, Ethiopia. Zone 4B, hot and very humid. 30-degree temperature will be there. 75% relative humidity, like India, Singapore, Zimbabwe, Thailand. These are some of the examples. So by using those conditions, we perform the stability study before marketing any product into the drug or commercial use. So there are three types of stability testing: long-term stability testing, intermediate stability testing, and accelerated stability testing. In long-term stability testing, we use the normal storage conditions, like normally our temperature, especially for zone 4B, if you take, it is 30 degrees centigrade, 75% relative humidity. So that temperature is called as a long-term storage condition. So in that long-term storage conditions, the developed products are stored at different periods. They are tested for the identity, purity, presence of impurities like degradation of products. So similarly, intermediate stability, that is the condition between the accelerated stability, long-term stability. This is the stability condition which is designed to moderately increase the rate of degradation. Accelerated degradation, irrespective of the zone, it may be zone 1, 2, 3, or 4A, 4B. So its temperature is 40 degrees centigrade, 75% relative humidity. So generally, accelerated stability studies, 3 months, 6 months, it will be performed. So based on this, they assign the expiry date of drug products. So here in this table, you can see storage conditions, long-term storage conditions, intermediate storage conditions, accelerated storage conditions for long-term stability, intermediate stability, accelerated stability. Here, majorly, each country having their own guidelines. Apart from that, internationally, International Council on Harmonization, ICH recommends the storage conditions for zone 1 and zone 2. These are the conditions. And for other areas, WHO, World Health Organization, it is an international organization. It recommends the storage conditions not only for zone 1 and 2, rest of the world, zone 3 and zone 4. Here, those conditions you can see. So the particular country, whichever the region they come, they follow those conditions for testing the stability. So the main aim of the stability testing provide evidence how the quality of the drug products varies with the time. So over the time, whether the quality remains the same or not, for checking that, stability testing is done. For establishing the shelf life for the drug product, shelf life or expiry date, determine the recommended storage conditions, where to store, whether to store at a room temperature or whether to store at a refrigerator or a freezer, and whether protect it from light, determine the container closure system, amber colored bottle or otherwise aluminum blister package. For different kind of packaging system, the stability testing is required. So as I mentioned earlier, stability testing is necessary to see, to check whether is there any chemical degradation. If there is a chemical degradation, the active consistent concentration that will decrease in our original formulation. So because of that, it may not be effective. Apart from that, toxic degradation products may be formed due to chemical degradation. So this is the background about degradation products and stability testing. So then, how we predict these degradation products? So here it is a practical strategy. So initially we do the literature review. So in literature, if somebody did it, we may get. So then in literature review, most of the new drug molecules, we don't get this data. Even some of the existing drug molecules also, sometimes what happens, this data, it is a, so this data that cannot be shared by a particular company. So then initially we see the literature, there we see the chemical structure, solubility, etc. data is available. So then we use the prediction of degradation impurities for that. The Zenith software is used. By using the Zenith software, you can predict the drug degradation products. So from that, we perform the degradation experiments. So once experiments is done, so then they're separated by using HPLC. So by using HPLC, these degradation experiments are optimized. So then these degradation impurities or degradation products, they are identified by using LCMS and NMR. So then once their structures are identified, so then we go for the in silico toxicity. In silico toxicity by using softwares. So once some toxicity is observed in the softwares, then we go for the animal testing. So here you can see the experimental design of degradation studies. So typically we do hydrolysis, because if you take our Earth, around 71% of our Earth atmosphere, it contains a moisture. Water, 71% of our Earth is occupied with water, and the entire our environment, it will be having the moisture. So because of that, most of the drug substances, they're prone to hydrolysis. So for testing the hydrolysis, we use acid degradation, acid hydrolysis, base hydrolysis, and solvent hydrolysis, typically water and some organic solvent. Here condition, we use 70 degrees centigrade temperature, and we do refluxing. We use typically 0.1 normal to 5 normal hydrochloric acid, or 0.1 normal to 5 normal sodium hydroxide, and the drug substance is degraded by using these acid or bases at 70 degrees centigrade. Similarly, peroxide degradation. So there we use hydrogen peroxide. Generally at room temperature we do. Similarly, UV light, visible light. These are the different strengths of these lights. UV light 200 watt-hour per square meter. Visible light 1 million lux hours. So at this strength, these are drug substances or formulations, they're exposed. So similarly, thermal, here a high temperature, generally, it depends upon the melting point, 60 degrees centigrade, 70, 80, at these 3 degrees, we subject the drug and we observe the degradation products. So this is all solution state. As you can prepare a solution of the drug sample or a formulation solution, and these different types of degradation are performed. Apart from that, even solid state also we can do solid state degradation. Here for acid, we use some citric acid, boric acid, base, sodium bicarbonate. We mix with them. The drug substance or formulation is mixed with them, and it is stored at 40 degrees centigrade, 75% relative humidity, and it is checked whether any degradation products are forming. Similarly, these substances are exposed to oxygen. These substances are kept in some beaker or a Petri plate in so then it is sealed, and it is checked at different time intervals. Similarly, these same solid substances of API or formulation, they are taken, they are exposed to UV light and visible light. And the same can be exposed to temperature alone. So then after that, so we go for the separation and HPLC for identification of the degradation products. So for this, literature is available. You can go through various papers published in the literature. Here I'm showing one, Singh and Bakshi. They have published one paper in pharmaceutical technology in the year 2000. So from there you can see all these conditions. So then now coming to these softwares, we have Zenith software with us. Zenith, this software is used for predicting the force degradation. So as I mentioned earlier, for generating these degradation products, it will take a lot of time. For example, if you take long-term stability testing, so generally long-term stability testing, we test it at 3 months, 6 months, 9 months, 12 months. So then 18 months, 24, 36. So first year every 3 months, second year every 6 months. Afterward, every year it is tested. So it will take almost 1 year, 2 years for predicting all these degradation impurities or degradation products. Similarly, if you go with the accelerated stability testing, there 6 months time it will take. So that is why what we do force degradation. In force degradation, what we do, we forcibly degrade the drug substances by using like one normal hydrochloric acid, one normal sodium hydroxide, hydrogen peroxide, similarly temperature, oxygen, even sometimes autoclaving also. So here we just take 1 day time. Generally, acid degradation we do 1 hour to 1 day. Within 1 day, you can get all these degradation products there in the force degradation studies. So then, apart from the force degradation studies, if you use this software, you can predict what type of degradation products are available. So before conducting the force degradation study, so first we collect the structure of the drug. So then we go for the force degradation pathway prediction by using a Zenith. So this Zenith, what it will do, it is a software. It will take the existing knowledge. So there are so many chemical reactions are reported in the literature. So from the literature, it will apply those chemical reactions. From that, it will predict the degradation products or degradation impurities. Nitrosamines also it can predict, and it will give likelihood scores whether how much whether it is sure, whether it is possible or not. Even with various excipients also it will predict. Generally in our formulation, not only drug substance, along with our excipients also will be there. So because of presence of excipients, sometimes what happens, this drug substance may interact with the excipient and it will form a degradation product. So this Zenith software, it is continuously improved. Regular updates will be done. So day by day, this software is also incorporating more and more chemical reactions. So let us see how this software works. So as I mentioned earlier, for predicting the degradation impurities, we can go for the long-term stability testing, accelerated stability testing, so then force degradation. These three are the generally performed. Here, Zenith, here take an example of Zenith. Zenith, if you apply on a drug substance, it is predicting 14 degradation products. Here this yellow one, it is nothing but it is the number of degradation products predicted by the Zenith. So then when you do forced degradation, only 9 degradation products are obtained. This orange color you can see. So A to I, around 9 degradation products, they practically obtained when this drug substance is treated with forced degradation conditions. So then when it is subjected to accelerated stability testing conditions, only 5 degradation products are obtained. When the same substance is actually done long-term stability testing, only 4 degradation products are obtained. So then you may ask, sir, then why to do these forced degradation studies, all these studies, if you are getting only 3 degradation products? Because we don't know which of these degradation products they appear in the final condition. Normal storage condition, A to I, which degradation product is formed, we don't know. As I mentioned earlier, it takes a lot of time. After 1 year, after 2 years, after 3 years, some degradation product is observed. So that is why, in order to predict at the early stage, we perform these degradations. So this is the use of Zenith. So then I will show you how this Zenith is going to work. So this is the Zenith software, how it looks like. If you click on the icon, it will give a dialog box like this. This is the Zenith. Here you have to enter your structure. Please click the edit button to add a query structure. Here you can click and add the structure, or otherwise what you can do, you can paste the structure also here. It won't accept directly ChemDraw. Here first what here you see, import the structure. Here type here or copy and paste here. Different formats are there: XML format, Inky format, MOL format, CDX format. Different formats are there. So generally what we do, we go for the smileys. So this is the upadacitinib drug substance. So for this, if you type in ChemDraw, ChemDraw will give you these smileys. Smileys are nothing but it is a simple notion. For example, if you want to draw a ethane, CC means ethane. CCC means propane. So that is the language of smileys. So this is the smiley for the structure. Once a smiley is created, so then that smiley is pasted here. Once a smiley is pasted here, you will get a structure. So this is the Zenith. In the Zenith software, now we have entered the structure. Then here you can see add excipients. Excipients also you can add. And here settings button is there. In that settings, you can give different conditions for predicting the degradation. So after giving the condition, this is the run button. In the run button, if you press, immediately it will give the degradation products. Let us see. Here, in the previous slide, in the settings, if you click on this, so then you will get the conditions. Here, temperature 70 degrees centigrade, pH 12 for basic degradation, pH 12. Here different pHs are there. You can mention pH 1, 2, 3 like that for acid degradation. For base degradation 12, temperature 70. At 70 degrees centigrade, I am going to subject the drug substance for reflux by using a base pH 12. And here solvent is a water. So then apply, in the apply button on the right side bottom, you can see apply. If you click on the apply, then you will get the results like this. Here you see, this is the drug substance. Sorry, you may not able to see all these structures. In the software, it will look like this only. It is a light in color. Here it had given primary degradation products. These are the primary degradation products D1, D2, D3, D4. And these are the secondary degradation products it has given. So here score is there, 900, 900, 700. So like that, you can predict the degradation products by using different conditions. Here you can use the different conditions, different pH you can use, and you can predict the degradation products. So here you can see the scores. 0 to 1000. This is software generally give 0 to 1000. Here you can see the possibility. If you have a score like 199, it is not possible, very unlikely. So above 500, there is a chance to generate, but above 900, so very likely. If it is showing a 1000 score, it is definitely it will form. So in this example, upadacitinib, that drug we subjected for this Zenith analysis. In acid hydrolysis, it has shown 3 degradation impurities. Similarly, in base hydrolysis, it has shown 4 degradation impurity with a prediction score of 900. As I mentioned earlier, so whatever Zenith predicts, sometimes you may get all, sometimes you may not get all. Sometimes you may get only a few degradation products. So here I'm showing the one case study from our lab. So we did the post degradation on upadacitinib. This paper was recently published in Journal of Chromatography B. Here you can see these are the chromatograms. Upadacitinib drug standard, acid hydrolysis, base hydrolysis. As I mentioned earlier, acid and base hydrolysis, it has shown 3 and 4 degradation impurities. But when we did it in lab, acidic hydrolysis, basic hydrolysis, we didn't get anything. But in oxidation, we got these are the impurities, 4 impurities with hydrogen peroxide, and this free radical initiator, isobutyronitride, one chemical is there. So with this free radical initiator also we performed the oxidation. There we got 1, 2, 3, 4, 5, 6, 7 impurities we got. Similarly, Fenton's reagent is nothing but ferric chloride. With ferric chloride here we got 2 impurities. And in presence of UV light we got 3. But whereas visible light, thermal, we didn't get any degradation impurities. And here I highlighted with some peaks I have highlighted with the circle. So these peaks, they are predicted by Zenith software. U4, U6. Here I have shown only Zenith software results of this base hydrolysis. But here I have not presented all the results of Zenith. So Zenith oxidation, U4, U6 also predicted. These two are matching with the Zenith data. And similarly, U7, U11, these are matching with the Zenith predicted degradation impurities. So for identifying these impurities, here you can see these impurities, they are present in a very minute quantity. You can see small peaks when compared to a drug peak. They are present in very minute quantity. In order to identify them, only mass spectrometry is the best technique. You cannot identify by using NMR. NMR require minimum 2 to 3 mg. So initially for identification, mass spectrometry is very useful. Or otherwise, if you want to identify by using NMR, you have to increase their concentration. You have to increase the degradation conditions so that their concentrations are increased. So then you have to isolate by using preparative HPLC. So then you have to isolate those drug substances by preparative HPLC. Then you have to confirm its structure by NMR. Then we have LC-QTOF mass spectrometry, high resolution mass spectrometry. Here it gives the mass up to 4 decimal points. Here you see for a drug substance, it is a 381.1645. This is the molecular mass of our drug. This is the one of the impurity, U1 impurity. Here you see 291.1427. This mass spectrum is the impurities mass spectrum. Here you see 291.1424. 1427, here 1424. Almost the same mass, accurate mass it is giving. So from this mass, you can say that this impurity is only this one. It is no other impurity. Not only this, you can go for the fragmentation. Here you see, this is the fragmentation. This is the fragment mass, 223.1053. This is the experimental mass. This is theoretical masses on ChemDra. These are the experimental masses. You see, same mass. Here also same mass, 192.1128, 1131. First 2 decimal points, it is exactly matching. So by using mass spectrometry, LC-HRMS, we identify the degradation products. So these are the various degradation products identified by using mass spectrometry. So then you may ask, sir, by using mass spectrometry also we can identify. So then why we are going with the Zenith? So these degradation impurities mostly they're related to drug structure. So we can easily identify by using mass spectrometry. But the thing is, for identification of certain degradation impurities, we don't know what kind of a chemical reaction drug has undergone. So everybody, they may not be familiar with the chemistry. So that is why we use that software. In that software, for example, U7, U4, U6, U11, these 4 degradation products, they are predicted by the software. In the software, not only it will give the structure, it will also give the molecular mass. From that, immediately assign the structure easily. Or otherwise, here, sometimes what happens, for structural elucidation by using HRMS, sometimes it may be half an hour, sometimes it may be 1 week also. It will take a lot of time. So at least for these 4 degradation products, we can save the time. So these 4 degradation products, they are predicted by the Zenith. So we easily assign their structures. Remaining structures, we worked a lot. So then when the structure is over, once you have a structure, impurity structure, so now it is the time to go for the toxicity. So whether these degradation impurities, they have the toxicity. If they show the toxicity, then we have to control these impurities. So here, so generally toxicity, traditionally they do animal testing. So generally in animal testing, so it will take a lot of time, and it will also you have to take a lot of animals and ethical approvals. You have to follow all the guidelines. So then it also involves a lot of cost. So finally, so then what happens, whatever the study result, so sometimes that may be directly correlated with the human beings, sometimes it may not be correlated. So then another thing is also there, you know that 3R principle of animal toxicity testing: reduce, replace, refine. So that is why people, what they do initially, they do this toxicity testing by using in silico toxicity. Once toxicity is predicted and in silico models, so then they go with the cellular models. Then once the cellular studies it is okay, so then they go with the animal models. So that is the strategy generally followed. So now let us see the toxicity of these degradation impurities or degradation products. So for this, direct is there. It is the widely used software for predicting the toxicity. So it gives many toxicity endpoints like a hepatotoxicity, carcinogenicity, mutagenicity, etc., etc. It will give. It is based on the quantitative structural activity relationship. It gives the toxicity. Here you can see, if you go to the software, direct software, here you can see direct. This is direct, Sara, Meteor, Vitic. These 4 softwares are there. Each software having its own utility. Direct is for overall toxicity, different organ toxicity. Sara is for mutagenicity. Meteor, this software is for metabolites identification. So similarly, Vitic software for this genotoxicity database. So once after going to this software, direct prediction, here you have to click on this. So then you have to enter the chemical structure as mentioned earlier by using the INKI files or smileys. So this is the structure entered. This is the drug structure I have entered. Upadisitinib drug structure. Here you see it is showing the results. After entering the structure, here I will show you. Yeah. After entering the structure, here you have to click here. Here you can see small notepad like a symbol. Here you have to click. Immediately it will give the report in this notepad. You can get it in a Excel, or otherwise it will give the result in a PDF format also. Here you see these are the results. Okay. So mutagenicity in vitro, it will use different animal models, bacteria, mammal, rats, different animal models it will use, and it will give the results. So here, mutagenicity in vitro, inactive, alpha 2 mu globulin nephrotoxicity, impossible, skin sensitization, non-sensitizer, alpha-2 globulin nephrotoxicity in mammal, doubted. So this drug molecule, it is showing it is safe because it don't have any mutagenicity or nephrotoxicity, no other toxicity. So in a similar way, we subjected all these degradation products. Around 11 degradation products are there. Here, U1 to U11. So then U2, it is showing ERG channel inhibition. ERG channel means cardiotoxicity. It is saying that equivocal. Equivocal means there is a possibility. It may happen, it may not happen. Plausible. 50% possibility is there. Mutagenicity in vitro. Developmental toxicity possibility is there. Mutagenicity possibility, plausible possibility is there. Hepatotoxicity is there for U7. U10 skin sensitization is there. U11 also skin sensitization is there. These are the different toxicities. These degradation products are showing in the software. Next, mutagenicity for this another software, SARA. This previous results are these direct results, and these are the SARA results. Now SARA. So SARA, it predicts the genetic mutations. So it uses the artificial intelligence by the existing data, and it will predict the toxicity. Here you can see a drug substance. A drug substance, each individual fragment also it is giving. So what is the possibility of the toxicity? And the overall toxicity it will give. So overall, here you see mutagenicity in vitro negative with 100% confidence. For drug substance, it is a negative. Now you can see in a summary here, direct data is also there, SARA data is also there. Apart from that, ICH M7 that is related to this mutagenicity. According to ICH guideline also, by incorporating those guidelines also, it will give the result. So drug substance, it don't have any toxicity. But whereas for impurities, here you see, U4 impurity, it is having a mutagenicity, it is a possible. U6, it is a equivocal. U10, it is a equivocal. U11, it is a equivocal. So mutagenicity, it is showing for U4, U6, U10, U11. So among them, U4, it is showing more possibility. So this is about the toxicity prediction by using Derek and SARA. This Derek and SARA, they're commercially available softwares. We have to purchase those software. Those software may cost around 3 to 4 lakhs for 5 licenses. But whereas in the public domain, some free software is also available. So this is the Protax 3 software. It is available. Protax 3 software, it can also be used for predicting the toxicity. So this software, it works based on the molecular similarity. So it also, if you give the structure, it will predict the toxicity based on the molecular similarity, fragment propensity in a molecular particular fragment, whether because of that fragment, whether toxicity is there or not, machine learning, pharmacophores. Based on this, it will develop a models, it will train the models from the existing toxicity data. It will train the models, and from that it will predict the result. Here you can see if you go to internet by typing Protax, there you will get the Protax web page. So there, U4 structure, degradation impurity U4, it was inserted. So then here you see the prediction of the various toxicities. So whereas in the previous software, in SARA, we have seen mutagenicity possible. So in this, you can see mutagenicity inactive. It is showing inactive by the software. But whereas its score is 0.51, if its score is above 0.5 and above, so then it will show green. It is inactive. It is predicting this software. But here you see it is showing the respiratory toxicity active, respiratory toxicity and blood-brain barrier. The drug, the impurity can cross. So these are the different toxicities predicted by Protax 3. So far we have seen the various softwares. First, the software is Geneth for predicting the degradation impurities, and next is the Derek for toxicity of different organs. Then SARA for mutagenicity. Then Protax 3 for different Protax 3 freely available software for different organs and different pathways. Then these are some of the challenges still with these softwares: data quality, the accuracy of the computational predictions relies on quality and quantity of available data. So as more and more data is available from the public domain, so these softwares, they can build the more and more training models. From that easily they can give the they can predict the exact toxicity. Another thing, model also, you have to validate this model by using the known toxic substances. This model shall be thoroughly validated, and multi-scale data integration, combining data from various sources, can improve the predictive power. As I mentioned previously, in SARA it is showing it is a possible U4, but in Protax it is showing inactive. So that is why 3 to 4 different platforms you have to combine, and you can get the exact data. So AI-driven drug design, so then personalized medicine, tailoring drug formulations and doses based on the individual patient characteristics. Sometimes this drug's toxicity depends upon the individual persons also. So like we have seen one example, some people are slow escalators, some people are fast escalators. So different people may have different kind of metabolism. So that is why those things also have to taken into consideration before predicting this toxicity. And quantum computing, so which is that is also very useful for molecular simulations and enhancing the accuracy. So with this, my topic is over, and these are my lab team members. I have almost 10 PhD scholars and another 6 PG scholars. Since the last 10 years, I have been actively working in drug stability testing and degradation impurities. Apart from that, I am also working on nitrosamine impurities, extractables, leachables. Even I am also working on this herbal drug characterization and co-amorphous systems also. Recently I started working, and I'm also working on this metabolomics, proteomics. So these are the various areas of my work. And thank you for all for giving me this opportunity. Now I'm open to questions. Hello.
Good morning.
Thank you, sir.
Yeah, thank you, sir.
Yes, please ask the question.
Good morning, sir. This is Dr. Selvi from CL Mehta College of Pharmacy.
Ma'am.
It was so very good information, sir. You got a lot of very nice info. Sir, I just need some information regarding this proteomics. Sir, sir said that you are having you have done some work or you are all already into research into proteomics. So my interest is on genomics, sir. Now, how advantageous is proteomics when compared to genomics? I heard proteomics is far more better application oriented while considering personalized treatment. So I would want you to just brief me about the intricacies of proteomics. Sir.
Yeah, madam. Proteomics, you can generally we can collect from some particular disease tissue, madam. So
Okay,
which
sir.
proteins are differently expressed? From that we can identify the pathway.
Okay.
In which
So-
pathway?
Yes, sir.
Which pathway is getting affected? So for that pathway,
sir.
we can develop the drug molecules.
Okay. Sir, you mean to say that based on the genetic predisposition of an individual, the
Yeah, ma'am.
protein expressed as a biomarker will be changed. Based on
Yeah,
that, we can decide a drug delivery or drug, deciding a drug, which class or which type will be better. That's what you're telling, sir.
Yeah, madam.
You,
With
you-
different population, we take the samples
okay.
and we analyze them. Commonly, which protein is up-regulated, down-regulated. From that, we will get the information. Which pathway is affected.
Okay, sir. So you need to say that for this particular proteomics based studies, like we don't need the gene banking or genomic banking is not necessary, you're telling. Correct, sir?
Yeah, ma'am.
Okay,
Yeah,
because
ma'am.
when we do genomics, pharmacogenomics, we require a complete genomic profiling of an individual. But when you
Yeah,
do
ma'am.
proteomics, you say only a protein expression pathway is far more enough rather than gene banking. That will be more advantageous, I guess. Correct, sir?
Yeah, ma'am.
Yeah.
Yeah,
Thank you so much, sir. Thank you, sir.
Thank you, madam.
So, Gananadhamu, sir.
Yes, sir.
Thank you, sir, for accepting our invitation in your busy schedule. Thank you for sharing your expertise with us today. I hope the participants are learned a lot and benefited from today's sessions. Dear participant, you can ask any questions to the part.
Sir, I think Vaibhavi madam, she have raised. Madam, please.
Yes, sir. Yes.
Hello, sir.
Yeah, madam.
I want to ask that why we to do stability testing when the drug is reached into the market, because in case of PG project or when go for Ph.D. project, some data is available with them also, manufacturing company. So why there is need or why will be the significance? This question is repeatedly asked by the judges. So how we can?
Yeah, madam, I agree. So, madam, this is a proprietary data, madam. So, companies, they won't reveal this data.
Yes, sir.
So we do this stability testing. We make this data available to our regulators, madam, regulators and our public users, so that they are aware of the consequences of these impurities. No company they disclose their data, madam.
Yes, sir. Yes, sir. And one more question is, sir, can I ask?
Yeah, madam.
Sir, in combination, when we have the combination of, say, suppose two drugs, then there is need is that we have to separate stability testing on single API, or we have to do it in mixture.
Madam, both the ways is the best way, madam. Initially, we had to do separately. So then combinedly also you have to do. So sometimes what happens, whatever the conditions we are applying for one drug, those conditions may be too stringent for another drug.
Okay, sir.
So that is why in both the way, separately as a combination also you have to do, madam.
Okay, sir. Okay. And one more is that, sir, the Zenith software, it is a paid software, or it is available online?
Madam, this Janet, Sarah, Derek, they are the paid software, madam.
Okay.
But their cost is less, madam. Around 3 to 4 lakhs you can get it. ProTax software, that is a freely available software, madam.
Yes, sir. Thank you, sir.
Yeah,
Thank you so much.
Thank you, madam.
Thank you, sir. It is a wonderful session, and you have covered about degradation products introduction today, stability testing methods, stability testing types, experimental designs, and softwares like Janet software for forced degradation pathway prediction, case studies with your publications, current status of toxicity testing, and one more software, Derek, toxicity prediction software, SARA, mutagenicity prediction tool, ProTax 3 broad spectrum tool, challenge and the challenges and future directions of this today's session. And it is a very useful session to us, sir. And thank you, sir. And our principal sir will talk to you.
I have to ask. Just count
Gana sir.
Yes, sir.
Yeah, good afternoon, sir. And I'm Dr. Manjappa, Principal, Vasantidevi Patil Institute of Pharmacy, Kodoli, sir. And my personal thanks, on behalf of management, I convey my thanks and gratitude for accepting our invitation and delivering fantastic information for all researchers and faculties who want to excel in research. And what I believe, sir, I think it is need of the hour to have an information related to degradation products. Generally, I believe that almost 90% drugs may undergo degradation in one or the other conditions. And whatever you have explained here now, it is really, give some idea of predicting impurities before you go for before you handling the drug. Sometimes you told that information may not be available on the public, but some may be with the literature, but most of the drugs we may not get information. At least we can use these softwares for the drugs which we are handling. And instead of searching all over, at least I can predict more number of degradation products, which possible degradation products out of the drug you are handling. And really it is informative, sir, because many can ask, but as you said, sufficient data may not be available in the public sector. And moreover, sir, I think even though we are having data, I think when we are handling drug for various formulations, we do nanotechnology and we do conventional and advanced formulations. These containing diverse excipients we need to use. So there we don't have the data for identifying, possible impurities. So one thing you said, this is more need of the hour, like drug and excipient interaction resulting in some new products, new degradation products. So if these software can be used for such kind of interaction also, sir?
Yes, sir. In Janet, that option is there, sir.
Yeah.
So can I show you, sir, in the
Yes.
slide?
Yes, sir. Please, sir.
Sir, can you see, sir?
Yes, yes, yes.
Add excipients. This is there, sir.
Okay.
We can add excipients also, sir, for predicting that.
Okay.
Sir.
Yeah, we can add our excipient of our choice and we can check the interactions.
Yes, sir.
Yes. And sir, with the interaction, we can apply all different conditions like change in the pH, change in the temperature, that causing interaction between drug and those selected excipients.
Yes, sir. Yes, sir.
Okay.
We can change the solvent, temperature,
Yes, yes, yes,
pH.
yes, yes.
Many simultaneously we can apply, sir.
Yes, yes, sir. Yes, sir. Exactly.
Like this, sir. You can see oxygen, some metal, radical initiator,
Yes, yes,
light.
yes. Because when we use excipient, no, sir, they
Sir.
have different functional groups. They change pH of the formulation, final solutions. Some likewise. So I think this software will help us a lot. I think.
Yes, sir.
Once again, sir, thank you for accepting our invitation and
Yes, sir.
delivering
Thank you, Vanjapan, sir.
and delivering excellent information for all of us, sir.
Yes, sir. Thank you, sir. Thank you, all the participants. Thank you. Can I leave, sir?
అడుగుతున్నదా?
Hello. Selvi ma'am, you are asking something. Query.
Please unmute yourself and ask the question. Sir, one more question.
Yes, sir.
Is there any free software for course degradation product prediction, sir? As you said, Genet software, Derek and Sara are paid software. And
Yes, sir.
Is there any other software for prediction of course degradation products?
As of now, we don't have, sir. For toxicity, we have, sir.
That is Protax, no, sir?
Protax.
Okay. Okay, I think there are no more questions.
Sham sir, you can proceed for
Thank you. Yes, sir. Thank you so much, sir, for accepting our invitation. On behalf of our management, Vasantidevi Patil Institute of Pharmacy, I would like to extend our sincere thanks for joining with us, sir, and sharing valuable speech.
Yes, sir. Yeah, I thank Principal sir and Shyam Sundar sir and Lakshman sir and the management of institution. Thank you.
I hope the participants felt very happy. But please stay for some while, sir. We are going for the valedictory ceremony.
Okay.
And dear participants, we are requesting. Now we are going forward to the valedictory ceremony. Please stay for some while. Good afternoon, respected dignitaries, Honorable President Sri Yashwant Shikshan Prasarak Mandal, Kodoli, Padmaja Patil Madam, Honorable Secretary Jayant Patil Sir, Honorable Trustee Mr. Vinita Patil Madam, and Chief Convener Professor and Principal Dr. Arhelli Manjap Sir, Conveners I.K.A.C Coordinator and Vice Principal for Academics Dr. Gauri Shankar Sir, and Vice Principal for Admin Professor Vikram Poddar Sir, Coordinators Professor Utkarsh Nagvekar Sir and Mr. Rahul Kadam Sir. Registration Committee members Mr. Anusha Madam, Ms. Shailaja Patil Madam and Mr. Sheetal Desai Madam. And technical team members Mr. Shiv Prasad Dojet Sir and KCR Venkat Sir, and fellow faculty members and esteemed resource persons Dr. Divya Shanmugarajan, Dr. Jupi, Dr. Veera Venkat Satyanarayan Reddy, Dr. Vasantaraju, Dr. Pallavi Jamdagni, Dr. Gananadhamu Sir, and dear participants. I am Dr. Shyam Sundar, Head of the Department Pharmaceutical Chemistry. It is indeed a great privilege to deliver the valedictory address of this faculty development program on emerging research trends in pharmaceutical sciences. Over the past few days, we had the rare opportunity to engage in exploring cutting-edge research insights and consider informative ideas that are shaping the future of pharmaceutical sciences. From the session's beginning, significance of toxicity prediction and drug safety using AML, leveraging in silico drug repurposing for advancements in drug discovery, smart formulations, AI-based drug delivery systems, redefining analytical practices using green chemistry, challenges and opportunities, Informatics, a novel tool in Ayurveda research, and in silico prediction of drug prediction products and their toxicity. Each session has been a window into the future of our field. The program has not only updated our knowledge, but has inspired us to rethink how we approach research and innovation. In an era when pharmaceutical sciences are central to addressing global health challenges, programs like these play a pivotal role in empowering academicians and researchers to stay ahead of the curve. We are especially grateful to our eminent speakers and domain experts who generously shared their wealth of experience and insights into both foundational and emerging areas of research. Their contributions have added immense value and will undoubtedly influence how we approach our work moving forward. I express our deepest gratitude to our respected Principal Sir, Dr. Arhelli Manjapa Sir, and Vice Principal Dr. Gauri Shankar Sir, and Professor Vikram Poddar Sir, whose unwavering support, encouragement, and trust made this faculty development program a grand success. From the very beginning to the conclusion, your guidance and full access to institutional resources empowered us to organize this event smoothly and effectively. Without your constant backing, this program would not have reached such heights. Thank you so much, sir, for being the main pillars of support for this initiative. I would like to extend my sincere appreciation to the Mr. Sanusha ma'am for delivering such a dignified and well-articulated introduction of our esteemed guest speakers. Your ability to highlight the speakers' accomplishments so concisely and meaningfully added significant value to the sessions. Thank you, ma'am, for your outstanding support. I would like to extend my heartfelt appreciation to Ms. Shailaja Patil, ma'am, a dedicated member of the registration committee, for your tireless efforts behind the scenes, from patiently responding to the participants' queries and attending the calls to promptly sending confirmation mails and ensuring smooth coordination. Your dedication was crucial to the success of this event. Thank you, ma'am, for your outstanding support. My heartfelt thanks to our outstanding technical team, Dr. KCL Venkat sir, Mr. Shiv Prasad Dojet sir, for their consistent dedication and behind the scenes excellence. Your proactive efforts in managing the online platform, fixing technical issues in real time, smoothly recording the sessions and uploading them to YouTube have been truly admirable. Your professionalism, patience, and technical skills ensured the event ran smoothly and remained accessible. I sincerely appreciate your hard work. My heartfelt thanks to all our fellow faculty members who extended their invaluable support by sharing the details of the program within their academic and professional circles. Your enthusiastic involvement played a key role in amplifying the reach of this event, significantly contributing the remarkable number of registrations and the overall success of the program. Your encouragement is truly appreciated. Most importantly, I wish to thank all the participants. Your enthusiasm, engagement, and willingness to learn made the journey collaborative and intellectually rewarding. A special word of appreciation goes to our host institution, Vasantidevi Patil Institute of Pharmacy, Kodoli, for providing the platform and support system necessary to make this FTP a smooth and enriching experience. As we conclude, let us carry forward the momentum what we have gained here with a strengthened sense of purpose and dedication. I wish you all ongoing success in your academic and research endeavors. Thank you once again, and best wishes to all of you. Thank you. So, dear participants, we will share now the feedback link. Please fill it and submit. And thank you very much for your positive feedback regarding this FDP. Thank you very much, one and all.
As a principal, I think my special thanks to all participants for your kind participation, for the knowledge, and hope you will cooperate in future as well with the further FDPs or any national level activities. And my special thanks to all Shamsundar sir and all organizing team for active participation and involving throughout the week. And my cordial thanks to all speakers. It's very difficult to get eminent researchers across the country, and we are fortunate to have such an eminent speakers from formulation to toxicity predictions and all the topics what Sham sir said. And my special thanks to all speakers for accepting our invitation and delivering their expertise, and that knowledge will help us all in the future research. So once again, thanks to all those who are involved directly or indirectly in this FDP, and my special thanks to all participants. Because of you all, this FDP became a success. Thanks. The videos are available because those who missed because of your academic scheduled or some other work, these videos have been recorded and they will be available at our college YouTube, and that link will be shared in the group so that you can keep it and you can go through anytime whenever you want to check it or you want to recall the knowledge.
Thank you, sir. Thank you, Principal, sir, for your valuable words. Now, with this, we are concluding the session 6 and FDP.
Dana sir.